Hi, my name is Christopher Anderson, and I'm studying environmental engineering at West Virginia University. Today I'm going to talk about my research with Dr. Emily Garner involving the composition and diversity of microbial communities in drinking water systems. And a drinking water system would include the process of water going from a source, such as an aquifer or a stream, then to a water treatment plant, and even further to a water tap in a place of business or a home. This is important because safe drinking water for the public is necessary to ensure human health. And so the more we know, the better. With increased population density, we have since gained the need for providing clean water through plumbing systems. You might consider then the opportunity for disease within these plumbing systems. Drinking water systems can be favorable for what are known as oligotrophic bacteria, which are bacteria that can thrive in low nutrient conditions, such as what you might see in plumbing, even with treatment and disinfection. A more recent example of a scenario involving waterborne illness might be the Flint, Michigan case in 2014, where Legionnaire's disease spread after the city had changed its water source. So as you can tell, the opportunity for pathogens or disease-carrying bacteria to pass through these systems and spread to the general public is and should be a focus for the scientific community. In order to understand and study the bacterial community in drinking water systems, we first have to determine what bacteria exists. So my research is focused primarily on both the composition and abundance of bacteria found in drinking water and biofilm samples. And biofilms are more or less a slimy layer of microbes that collect on wet surfaces rather than being suspended in the water. So using data from Dr. Garner's previous studies, that includes samples from 4 utilities in the West, Southwest, and Southeast United States, I have used bioinformatics software to take raw genetic data from these water samples, cluster that genetic data into groups with 99% similarity, and then compare these groups to an available database to determine the bacterial composition in each sample. So the analyses that I've completed are here in the results. And first, we have a taxonomic bar plot that shows the relative abundances of bacteria in each one of the samples on a 100% scale. Then you can see a box and whisker plot representing the alpha diversity of the samples separated by utility. The alpha diversity can tell us diversity on a local scale. So a higher index means that more bacteria types are present and at more even proportions. Lastly, we have a beta diversity analysis, which shows the diversity among multiple samples. And so the closer the two sample points are to each other on this plot, the more similar their overall bacterial structure is. And you might take note of the QR codes here that will allow you to play around with this plot a little more in 3 dimensions. You can either follow the steps to view this exact plot or look at other variations and trends available within our data set. Although research is currently ongoing, the primary objective for now, as mentioned, has been to establish the community of bacteria within the samples. And some preliminary findings were that a large part of the species were found to be proteobacteria, which we would like to look into further to see or maybe ask the question if certain non-pathogenic proteobacteria in a biofilm, for example, would provide an environment that is suitable for pathogenic proteobacteria to thrive. Also, the beta diversity showed that samples from the West and Southwest US were more closely aligned to each other than those from the Southeast, suggesting a physical or geographic factor could be involved. For the short time I've been researching this topic, much of the effort has been on getting results of what specifically exists within these samples. However, we hope to continue this research well into the fall and dive deeper into our analyses to see what specific factors are influencing the bacterial communities. With that, feel free to reach out to me at my email or within the comment sections for any questions you might have. And don't forget to play around with that QR code to see a multitude of possible trends and variables that we hope and we can continue to investigate moving forward.